All right, and let's go. All right, so this is certainly not a statistics presentation. This is intended as an overview. I am going to try to give some idea of what the statistical underpinnings are, why we get efficiency from certain things. But certainly if you'd like a nuts and bolts kind of presentation, I'm happy to come back anytime, and I'm happy to answer questions at the end of this. One thing to keep in mind is kind of the modern randomized clinical trial was created in 1946. Some aspects of this are wonderful. I mean, randomization is hugely important for generating good causal inferences. But certainly, the world didn't stop in 1946. There's lots of room for advancement. I've got pictures here of a computer and an operating room in 1946. And certainly clinical trials have advanced. And I'm going to talk just about some of that. There are lots of different ways clinical trials have advanced. The real problem that I want to discuss today is really a search problem. A notion that we're trying to investigate a condition, a disease, and we've got many possible therapies. And the question is, how do we search through that space given our finite resources? So this isn't a simple problem of, I want to know, does agent A work in a particular disease? I'm in the situation where I'm investigating COVID and there's literally dozens of possible treatments, and I need to explore all of them and find what works and what doesn't. So I'm talking about ways to make use of this kind of this broader problem, ways to increase the efficiency of standard trials, and these are going to combine a lot of old and new ideas. Just to keep in mind all the different things for COVID-19, I tried to kind of write out a list. We certainly heard a lot about HCQ at the beginning, still a little bit on that. Other kinds of antivirals, there's immune modulators, there's anticoagulation therapy, antibiotics, all kinds of things might be used in COVID. And I started this list and then it got very long very quickly, so I kind of condensed it on down. I want to talk about really three specific innovations. One of these is early stopping adaptive designs, and I'm going to talk about the vaccine trials. And then what I'm going to do is talk about platform trials. If you've heard of a trial called I-SPY 2, it's often used as in news articles, we want a trial like I-SPY 2. But I want to talk about what those do. And then I want to talk about remap trials, which are a particular kind of platform trial. And I want to talk about remap, CAP, and COVID. So again, all of this is going to be at a high level. I'm certainly not proving to you in any way that these are the right things, but I do want to convince you that this is a reasonable idea, and I'm asking for a bit of faith that we have spent a lot of time researching this. A review article came out over the summer that had about 300 references. So this is not just done haphazardly. I also want to talk about some of the potential obstacles here. So one of the unfortunate things that's been brought out in COVID is people are doing a lot of things badly and often attributing them to innovation. And, oh, this is innovative, when really it's just bad. And so I want to kind of give some idea of what you want to look for when evaluating these. So let me dive into stopping early. And I'm going to talk about the 4 COVID vaccine trials. All of these allow early stopping in some form. Pfizer is the most aggressive. Their CEO has, in my opinion, been a little too vocal about their status of their trial. I prefer they let the trial run in peace. But Pfizer's the most aggressive about stopping their trial early, but really all of them, Janssen, Moderna, AstraZeneca, they all have possibilities for stopping early for success. In some news articles, these have been criticized as shortcuts. And so I want to talk a little bit about what should we really worry about, what should we not worry about. And I want to emphasize that we need to consider this both from a prospect of safety and efficacy. And I'm going to spend most of my time on efficacy because that's what the primary analysis is, but then I'll come back to safety at the end of this. So again, don't take this as a full endorsement of each of the individual trials and every rule in them, just that I am going to defend that this is a reasonable idea. So I had a client discussion a while back, and we were talking about, they needed a sample size of about 200. And they asked me the question that they said, is this when the result becomes significant? And I think they had it in their mind that we just accumulated evidence and somehow I solved that, imagine an airplane coming out of the clouds. And at 199, we're in the clouds, everything looks white and we can't see what's going on. And at 200, the clouds part and suddenly we see the answer. And certainly that's just not the way the world works. When you're a statistician, you get to say that, statistics means never saying you're certain. Random data is random. And so when the clouds part for us is actually not easy to predict in advance. So this gets at that notion when we talk about stopping early or shortcuts. It has the implication that the 150, 160 events, all of these vaccine trials, they're waiting for 150, 160 infections to occur. And so it has an implication if we say stopping early, there's a notion that 150, 160 is the right size. And so I want to talk about that 150, 160, where it comes from and why you can argue that it's actually really late and you should try to be a little more efficient. I'm going to focus primarily on the methods that are used in the Janssen and Pfizer trials. They're a bit simpler, but the intuition is going to hold generally. The way a vaccine trial works, we vaccinate a large number of people. We follow them up over time. And what we're looking for is infections to occur. And the trials like Janssen and Pfizer, what they do is they say, okay, when a certain number of events happen, we're going to look at the split between the events. How many of the infections are in the control arm? How many are in the treatment arm? And certainly if the vaccine works, we expect that there are a limited number of infections in the treatment arm. So most of our infections, if they're in the control, that's a good sign. Statistically, we know a lot about what to expect here. If the vaccine had no efficacy whatsoever, VE is vaccine efficacy, then we would expect half of them to be in each arm, 50% control, 50% treatment. If the vaccine goes up to 30% effective, and keep in mind, 30 is viewed as the null hypothesis for a vaccine trial. You got to show that you're better than 30. And ideally, you want 50 here is what the FDA is looking for example. But at 30% effective, we will have eliminated 30% of the infections on the vaccine arm. So the vaccine arm should have 70% of the placebo arm events. And so if we look at those together, 100% of the events in control, we haven't done anything to it. 70% of the events are in the treatment arm. And so when we look at that split, it should go down to about 41%. The general formula here is, and we don't need this, but just for reference, the proportion in the vaccine arm, 1 minus VE over 2 minus VE, fairly standard formula in this area. So really what we're looking for is a good vaccine, the split, we're going to have less than 41% on the vaccine arm, and ideally much lower than that. Okay, so how do we design a trial with that? Well, we want to get an idea of the kind of range of variability that a dud vaccine would do. So each of these curves here, these are all possible paths from a vaccine that only has 30% vaccine efficacy. So you can see, for example, some of these look really bad early. It might actually look like they're harmful. This horizontal line is what we're looking for. And remember, good vaccines have a small number of infections on the treatment arm. So some of these look bad early. Some of these vaccines look pretty good early. So you can see this is about maybe 25 events in. Here's a possible path that actually had about 18% in the vaccine arm. So lots of variability here. The early results could be anywhere from, 20 to 60. As we go forward, all of these paths start to coalesce around that 41%. So the nice thing about statistics is as you observe more data, everything becomes more certain. So a good vaccine, one thing we want to make sure is that we distinguish it from this. So we want to make sure, for example, that if we're going to claim efficacy, that what we're going to get, we have to be below this range of variability. We need our data to be underneath this because we don't want to confuse it with these random paths that a bad vaccine might take. So we need to be down here, and this is where we get into notions like type 1 error. We need to be below the 2.5 percentile. That's where our 2.5% alpha is coming. So essentially, if the vaccine trials are looking for 150 events, you of course do out all the math, but you look here, find the 2.5 percentile, and if you're below this, you win, you claim efficacy. In contrast, a good vaccine, most of these trials are powered for about 60%. You would expect them to look better. So all of these green paths are a 60% vaccine efficacy. So most of them are below the black. The vaccine's better. So that's good. But you can see there's overlap here. So clearly there's a fair amount of overlap, particularly early. And so one of the reasons that we wouldn't just pick, say, 32 events, so Pfizer has a look at 32 events in their protocol, but the reason that they don't just stop there, the reason that the trial doesn't end, is that this overlap, if the data's in this region, I can't tell you if the vaccine works or not. The vaccines that don't work might be here, the vaccines that do work might be here. So this, too much overlap. So why do we actually pick 150? And the reason that we do that is we're waiting, and I know a lot of this is old hat to people on here, but I want to kind of lay out all this so I can add more to this graph. At 150 events, what has happened is we're looking for this to have separated. So these, the black curves are now up here, the green curves are down here. Obviously, we do this on a percentile basis, controlling our type 1 error, 2.5%, and we want to control our type 2 error as well. So we're waiting for these to separate enough that we feel confident we're going to be able to tell the answer. If the vaccine ends up down here, yes, it works. If the vaccine ends up here, then no, it doesn't. So we're waiting for these to separate. If you can only look once, that's a good strategy, because we certainly don't want to go halfway through the trial, go to 60 events, end up with something inconclusive, and then we go, oh, what can we do? We can only look once. So really, we're buying an insurance policy. We're waiting for everything to separate. That takes a long time. And so we're saying, look, if we can only look once, we're going to wait a while. Going back to this cloud analogy, it's essentially saying, I can't tell you when the clouds are going to part. But I can tell you that they will have parted by the time we reach a certain point far into the future. And so instead of keeping our eyes open all the time and just waiting for us to move out of the clouds, what we're really doing is we're saying we're going to close our eyes, we're going to put the plane on autopilot, and at least we're confident that when we finally open our eyes again, the clouds will have parted and we'll have an answer. So that's why we wait to 150, 160. And in many respects, you can argue that this is pretty late because there's lots of times that we come out of the clouds earlier. And so you can see, for example, a lot of these green paths, they are firmly below the black curves earlier. So some of these green paths right here, there's no black curves in this area. These are pretty conclusive results in here, in here. You can also tell on the flip side that there are some ineffective, and I know these arrows kind of dip into the black, it's hard to see the points, but up here you can tell these are duds. These are ineffective vaccines early. Can you make mistakes doing this? Yes. So here's a green path that is kind of overlapping. I think there's no black paths in this area. So it's possible to make mistakes, but they're limited. And of course, if you go to the end, you can still make mistakes. If the vaccine works even better, so we had an 80%, I don't know quite how likely this is that we're going to get this lucky, but they separate even earlier. So these are the blue paths, and you can see they're pretty distinct all the way down 25 events or so. So really, you know, all of this waiting out to 150, we're trying to guarantee an answer, but we may have seen an answer earlier than that. So let me kind of skip this slide since I've already said it. These are the Pfizer splits that they need. This is just copied out of their protocol, which I am thankful that they have made public. They're doing an interim analysis at 32. And their success criteria, if that split is 6 in the vaccine, 26 in control, or better, then they're going to go ahead and declare efficacy. Now, I want to be careful about, they are not declaring safety, and that's what the real dispute, I think, ought to be here. But they will declare efficacy. They also have a futility rule. So if that split is 15 out and 17 or worse, then they will end up dropping for, anyway, they'll drop for futility. And so you can see the splits here at further interim analyses of 62, 92, 120, and they have a final analysis at 164. So if they can hit any of these splits, they win. And so what I've done on this graph is I've tried to put down where their success regions are. And so they can win at 32, they can win at 62, they can win at, my apologies, 92. So this is 92, 120, and then this is their final analysis. And so the important point here is that these have a collective 2.5% type 1 error. If they're talking about a vaccine that's at 30%, then it's not desirable, then these black curves, they don't get into these red regions. There's only a 2.5% chance that a black curve would hit any of them. So that kind of error control, they get the same kind of error control in their trial that you would if you just went out and made one look late. So that 2.5% is maintained here. You certainly have to, a very important point is in order to verify that we have that 2.5% type 1 error control, you have to take into account that they've made multiple looks at the data. And there are lots of different formulas for this. Ironically, each one of these trials uses a different kind of calculation. They're all very related calculations. So they differ in the details, but they all demonstrate that they control the error rate, even accounting for the fact that they've looked multiple times. The kinds of things that you want to watch in this kind of trial, let me kind of pause for a second. I don't have a slide on this. But generally speaking, these kind of adaptive features, they will save you for success stopping. You probably save 20, 30% of the sample size. For futility stopping, you can save about 50% of the sample size or more. Futility is usually the more valuable thing to do in trials. I recognize for an academic audience that's a little awkward because grant agencies, you obviously don't want to give back your money. And so anyway, the granting agencies are struggling with this notion, but from my conversations with NHLBI, others, they are actively trying to facilitate this kind of activity where you can do this and move on to something else, or however, anyway, they're trying to figure out more options. The kind of things to watch here is you have to pre-specify all this. So this, let me go back a slide. This table is sitting in Pfizer's protocol and has been there before they enrolled a single patient. So they needed to say in advance what they're going to do. You cannot do ad hoc evaluation. It's uninterpretable. You can't compute error rates. It ends up being a mess. So for example, it's difficult to put this in the hands of a DSMB without guidance. Now, that's not saying the DSMB can't respond to safety risks, can't respond to signals that are so vast that they just have to stop for loss of equipoise, but general adaptive stopping that gets you efficiency gains, you need to pre-specify it. You need to make sure that you've done these kind of formulas to have careful control of your error rates. One of the consequences is the maximal sample size will go up. So Pfizer, if they don't win their early interims, they need to go to 164 events. Other trials might have to only go to 150. So typically you're taking a small risk of needing bigger and you're getting a larger opportunity to go smaller. But you need to keep in mind that if you want to control your error rates, you've got to do this right. You also need to consider other outcomes here. The most important thing here is safety. So Pfizer's early interim, if they get a declaration of efficacy, I trust that they're efficacious. Not sure they've fully demonstrated durability of immunity, but they're efficacious for the time frame they studied. But what they haven't demonstrated necessarily is safety. And so all this discussion, if you've been keeping up with the news, of the FDA asking for a certain amount of 2-month follow-up on patients, that may preclude Pfizer from doing their early interims. And I'm certainly not privy to those discussions that are going on right now. I wouldn't be surprised if Pfizer has their events and is currently discussing with the FDA, how much safety do you need? I don't know that, but I can just, from experience on doing these, that kind of conversation could be taking place. You also have to be careful here if you stop early for futility. You don't want to stop a trial too early that might recover and go on to success. So really aggressive futility stopping can lose power. And so it's important that you've done that calculation and you've said, look, I am only stopping things that would have failed anyway. And so that would limit your loss of power. But if you're reading through this, you should be seeing all these things. Here is the power loss from early stopping. Here is the needed increase in maximal sample size. Here is our expected reduction in sample size, in expectation, in average performance. And a very careful thing that they have controlled their error rates and taken into account all of their looks. All right, so that was early stopping. As I said, gains, I've got another short course that we did for NHLBI. Just doing that could potentially allow a granting agency to fund 50% more trials. So if you've ever been right below the pay line, you should be very upset that all the trials ahead of you did not do this, because potentially there would have been enough money for you. All right, so that's early stopping. Let me move on to platform trials. I'm building here. Platform trials are certainly going to use these early stopping ideas. In Alzheimer's, so let's kind of think of the society problem. There's been at least 25 major failed trials. If you really look, there's probably 100, maybe 200 failed trials now in Alzheimer's, but they're not all huge. They all have different sample sizes. Some are bigger, some are smaller. For simplicity, let's just assume that each of them had 1,000 per arm. So that means as a society, we've enrolled 25,000 control subjects, and we've enrolled 1,000 subjects each on 25 experimental therapies. So this was how we searched the space of those 25 potential treatments. So we did a whole lot of controls and a little bit for each one of the treatments. That's a little inefficient. So if we were really going to investigate 25 things, we would not decide that we want to know tons about doing nothing and a relatively small amount about every treatment. So the question is there a better way to do this? And platform trials are aimed at the idea of investigating multiple therapies at once. And so you'll hear this term master protocol. This refers to really a protocol without any treatment names. So if we're studying Alzheimer's, we would define things like our endpoint is going to be ADAS-Cog. It might have some functional measures, all of the kind of endpoints that happen in Alzheimer's. We would define all our procedures. We would say, we're going to collect data at 3 months, 6 months, 9 months, our final endpoints at 18 or 24, whatever might be the right thing for this particular master. But all of these kind of general procedures are going to be written down, but there's no drug name. So it's the kind of idea that if I were investigating novel agent A, I would do this. And if I were investigating novel agent B, I would do basically the same thing. The trial is going to incorporate multiple treatments, and each one is going to get its own, what's usually called a drug appendix. And so these appendices, then that's where all the details of the individual drugs come in. So it would say, this is the individual drug. Here are all the details of the chemistry. Here's everything we know about safety, all the things that would be very specific. And it's possible, for example, there may be some exceptions to the master protocol rules. Often these take place in terms of patient exclusions. We designed one of these for the Ebola outbreak in West Africa, and each of the individual arms, they could have an exclusion on whether or not they would enroll children and whether or not they would enroll pregnant women. So every one of them had to make a specific choice on that. You could also put in the drug appendix, if one of them you're worried about liver toxicities, then there may be a statement that says we're going to collect extra labs to make sure we're monitoring this specific known issue with this drug. Ideally, these differences are limited, but they don't break anything if there's a few of them. A platform trial is a very specific form of a master protocol. It's defined by the treatments. Essentially, we often call this a cohesive inferential scheme. The treatments are typically entering and leaving the trial. There's 4 to 6 treatments at any given time. Might be more, might be less, depending on the individual trials. And I want to kind of indicate there's going to be randomization between all the arms. So we are going to be not just, this isn't your kind of, there's a lot of cooperative groups where they manage a patient stream and multiple trials happen in that group. Certainly if you have one of those, you're well on your way to doing this platform trial. But in those kind of settings, trial A is if you get into trial A, you get a randomization. You might be in trial B, you might be in trial C, and so forth. This is a situation where when you're randomized, you might get control, you might get A, you might get B, you might get C, and the randomization structure incorporates all of that. The key reason for this is because everything is randomized together, it facilitates the sharing of the control arm. Now, typically, I would recommend that you make your control arm bigger when you do this. So if you were talking about, say, 5 trials that you would enroll 100 each, so you've got, 100 trial 1, 100 trial 2, 100 trial 3 in terms of patients, 100 trial 4, 100 trial 5, what you could do is you could enroll 250 patients to control, 150 to agent A, 150 to B, 150 to C. And so what you see is the individual comparisons. I've got 150 treated subjects and I've got 250 controls. I dramatically increase the sample size, but I'm not skimping. I'm actually increasing it. And you could go somewhere in between to actually reduce the overall sample size. These trials are often intended as perpetual. So it's intended that treatments will leave over time and new ones will come in. Kind of an example of this. So this might be a paradigm where you start off where you're enrolling a control and you're enrolling 3 different arms. The WHO Solidarity trial, for example, it was enrolling 4 arms and control at the same time. And patients were randomized in that scheme. Solidarity was not a full platform because they don't have the features I'm about to show. So you can add arms later. So arms D and E could come in at some point. You can, all of the adaptive features, so arm C could drop for harm. A lot of times these are set up so there's a certain number of slots in the trial. We're always enrolling 3 arms, and we wouldn't bring in a new arm until one of the current arms stops, either for futility or for efficacy. You could add combinations. So if we find out A works and D works, we could try them together. This is a bit rarer with one exception, which I'm going to talk about in a moment. But that tends to be a more isolated feature, but you can do it. Certainly you can stop arms for superiority. And while this is disruptive, it is not catastrophic. If an arm is better, you could actually switch the control out. So I-SPY 2 is a breast cancer trial that's been running about a decade, and its control has changed. Pertuzumab was approved for certain kinds of breast cancer, and pertuzumab became the control arm in I-SPY. It had the advantage that it had actually been through I-SPY as an experimental arm, so we already knew a fair amount about it. So all of these are possible. You don't need to do all of these, but the primary thing that defines a platform trial is you are investigating multiple things at once, and you are randomizing among all of them to try to get this cohesive inferential structure. So to give an idea about why we get efficiency gains here, I'm going to do this in the context of an epidemic. So certainly we have one of those going on. This is an old example that we've shown on a few slides, and this was more motivated for the Ebola outbreak. Suppose that standard of care has 30% survival. We certainly want to improve on that. We want to explore a lot of novel agents, and unfortunately, most of them don't work. So 10% of them work, they improve survival to 50, 90% don't, they leave survival where it is. So this is just a simple example. I'm laying out, this is the state of the world. And now the question is, I don't know which of the novel agents work. I know most of them don't, but I don't know which one's which. And so the question is, how do I search through a large number of novel agents quickly in order to find something that works? And so, within a traditional trial paradigm, we might investigate them one at a time. So I have so many resources. I'm going to grab, if you imagine COVID in March, I'm going to run a trial on HCQ. If HCQ doesn't work, then I'm going to go, okay, next one up is remdesivir. I'll run a trial. If remdesivir works, I wave a flag and say, fantastic. If it doesn't, I go on to lopinavir. And if that doesn't work, I go on to something else, and on and on and on through this. So I'm running one trial at a time, just in sequence. And what I'm trying to figure out is, how long do I have to do this? How many patients do I have to investigate before I found, the needle in the haystack, the one of the novel agents that does indeed work? And so there's a paper that has all these results out in clinical trials. I'm just going to summarize. This kind of study, you need an average of about 2,000 patients. So 1,966, these are simulations that have some simulation error attached. Remember, 10% of the agents work, so we really need to look at, on average, about 10 of them before we actually find one. We might get lucky, we might not get lucky, so it could be higher or lower. But on average, about 1,966 patients, 2,000-ish. Another prospect is to just start moving towards this idea of sharing controls. So I'm just trying to build up the aspects of the platform. So I'm going to investigate 5 at once. So instead of doing a 100-100 trial, checking the results and moving on to the next one, I'm going to do 600 patient trials, and I'm going to do 100 controls and 100 to each agent. As I said, my personal preference is to enroll a few more controls. I think that's a good way of utilizing your savings to maintain a bit more robustness. But this example, 100 each. And then at the end of this, I'm going to say, well, do any of the 5 work? And if something did, I'm going to stop the process. Otherwise, I'm going to run another trial with 5 new agents. Now, typically in this situation, if you're a statistical kind of person, you should immediately be thinking, oh, you're testing multiple things. Remember, we're always testing multiple things from the society view. There's no type 1 error penalty if you do separate trials. These are judged by the FDA on similar reasoning. Is if you could have tested them separately, we realize that you're doing this as a search agent, you're efficiently running all these trials. So the type 1 error is per agent. All right, so this gets you down to 1,528. And the savings here is through the sharing of the control arm. So let me kind of back up a few slides. What we're really doing, and I guess it's several slides, this Alzheimer's example, we're essentially saying we know too much about control. And so by sharing it, we get some savings, 1,528. We typically combine this with the same kind of adaptive ideas that I just talked about for the vaccine trials. So we allow drugs to stop early. Here, the primary thing that stopping early gets you is futility. Most of the drugs don't work. So we're going to get most of our savings here just by dropping out dud drugs as quickly as we can. And so that drops down to 971. And again, I want to emphasize, none of this can be haphazard. This requires a lot of prep. If somebody does this and can't supply a lot of documentation to you that, these are our error rates, we've taken into account our accrual, we have an understanding of what our drug population looks like. If they can't tell you that, then they're going to end up in a bad place because they're going to get an ad hoc, uninterpretable result. But if you do this right, notice we've dropped the sample size in half. So, these are not small gains. These are massive gains. And finally, we get to this open platform. So what we're also going to do is anytime a drug stops for futility, we're going to bring another drug in immediately into this. So we always have 5 agents at a time versus control. Anything that drops is immediately replaced. And we're just waiting how long until we can finally find something that works. And so this is the open platform, and you can see this is down to 849. And there are a few other bells and whistles we can add to this to lower it even more. Those are situation specific on whether they make sense or not to add into this. Again, we have not lost in terms of error rates by doing this. And again, we've been careful to calibrate all of our decision rules here. So our type 1 error rate is still 2.2% per drug compared to 2.5% per drug. We actually have more power for individual drugs. One of the things that we've been able to do is by sharing the control arm, we get a little more accuracy out of this. So nothing's been sacrificed here. This is just making use of the savings. And really, there are three forms of savings here. There is sharing of the control arm. There's the benefits of stopping early when we know the answer. We don't want to stop early unless we know the answer. And the other thing is investing those savings forward. It's not compound interest. You don't get exponential growth, but this notion of any sample size that we save from futility, we're going to immediately use it to get another drug in. This is a challenging thing to set up. It is a longer setup. It is a more expensive setup. It is difficult to get the first agent into these trials. Most of the trials that do this have some kind of grant funding in order to make them happen. Some aspects of the trial are more complex. Informed consent is typically two-tiered. Blinding. There are methods I'm not going to discuss here for maintaining blinds among multiple agents, but that is typically more complicated. You need to think through all these issues. The advantage to them is because they have these, they're so much more efficient. Once you can get them up and running, all the other agents go in and go, well, if you're already enrolling, say, 3 or 4 of these, I have a tremendous incentive to come in later. So if you can get this warm base up, then usually what happens is it's easy to convince the 3rd, the 4th, the 5th person to come in. So it's kind of, if you build it, they will come, but you have to build a really hard thing first. These are all supported by regulators, academic groups. So this is Janet Woodcock. She is the head of the Center for Drugs at the FDA. Realistically, the person who is kind of in charge of drug development. Lissa Levange was the head statistician at FDA CDER for a while. She is no longer. She's gone back to North Carolina. But this is a regulatory perspective in New England Journal of Medicine. This is a article in Nature Reviews Drug Discovery. This one has a lot more details. So if you want to read more, I'd recommend this article. And so this also, Janet Woodcock is part of this Adaptive Trials Coalition. I'm on this. If you're a mathematician, I have my Erdish number through this paper. This also, the CTTI is a group from Duke, affiliated with Duke, has also connections to FDA, and they've just completed a whole thing on master protocol studies, and there's a series of white papers. I was part of this as well. So some of this is touting mine. This is theirs. So anyway, this is certainly not, this is encouraged in the US. It is a little more controversial in Europe. I can talk about that if somebody would like to know. It is not just an idea. These trials are actually enrolling. iSpy 2 has been going for a decade. GBM Agile is currently enrolling at a number of sites. This is another trial, PanCAN's Precision Promise in pancreatic cancer. And there is the Healy ALS platform trial, of which UK is a site, and I think I saw the site PI's name on the participant list. So thank you for being here. So these are all platform trials that are have FDA approval, have been going through enrolling patients, and in the case of I-SPY, it's had about 15 drugs already go through. These other trials have started more recently. All right, again, things to watch here. These require extensive preparation. You need to do all this work, make sure that they show you the documentation for this. It should be in the supplementary material and papers and everything else. And then the final thing I want to talk about here is remap trials. This is a quicker topic to close on. This is a very particular kind of platform. The remap stands for randomized, so randomization is good for lots of things. These are embedded trials. They're embedded within the healthcare system. There is consent, but they use EHRs and so on. That's to allow better enrollment. They're cheaper. Multifactorial, so I'll talk about this in a minute. And they are adaptive platforms. The remap trials, the M for multifactorial. A factorial experiment is a really old idea in experimentation, and it says if you want to investigate agents A and B, and they can be combined, you could do one trial in A versus not A and another trial in B versus not B, but it's actually more efficient to do everything at once and to have 4 arms and do placebo, A only, B only, and the combination. And so this is a basic factorial where you cross treatments. Factorial designs are more efficient. If the treatments don't interact, the factorial experiment is essentially free. You learn about both for the cost of one. That's usually unintuitive, but I'm sure Heather would be happy to go through all the details. If the treatments do interact with each other, then running the separate trials, you can't estimate the interaction. Whereas with the combination, you can. That trial may need to be bigger. It's not free, but it's a key part of the scientific picture. In COVID, what we're seeing right now is really an unfortunate aspect of competition. So I've been in meetings, we talked about the active trials. I'm not in those, actually I'm not in those meetings, but I do hear about them, we participate in them. But there's discussions where, you know, person A is running an antiviral trial, person B is running immune modulation, person C is running anticoagulation trials, and they start to discuss, well, none of us can enroll. And the reason they can't enroll is they're all competing for the same group of patients. These can be given in combination. So maybe not everything with everything, but lots of combinations are possible. And so this suggests that kind of factorial design. And so there is a trial enrolling on this that's called remap cap. You can go to, if you just Google remap cap, you can get to the website. It's enrolled 1,628 COVID patients right now. It was designed for just regular community-acquired pneumonia. So the total number of patients is a bit bigger. 267 active sites right now throughout the world, including US, Europe, New Zealand, Australia, Canada, and they are trying to expand to other places. And again, what this is trying to do is it's trying to take these different domains. So instead of running an antibiotic trial, an antiviral, and a steroid trial separately, we are assigning regimens. So a patient that comes in, you get an antibiotic. Now, remember, the antibiotic might be placebo. It might be nothing. But you get an antibiotic, you get an antiviral, and this is a bit dated. I'll talk about the steroids in a minute. But you could get steroids and you'd get randomized to a regimen. And then what we would do is we would investigate this as a factorial experiment. You can have domains and agents evolve over time. So we can do things like add new antivirals. You can add new domains. So remap cap now has an anticoagulation domain or is about to. I actually don't know if it's enrolling yet. And domains can finish. So I use this slide because they were pretty pictures. The steroid arm is actually not enrolling anymore. And that's because it has read out and they think there's a decision on the steroid arm. Now, this is actually an interesting decision because this was a combination of the remap cap info combined with the info from recovery and other studies. But the whole domain is now enrolling one way because it's viewed to be the best. So anyway, summary here. We certainly did not stop advancing experimentation in 1946. Obviously, tons of people are doing research in this, and I'm only talking about one of them. But the main point I want to make is considering only one trial is fairly limiting. We need to consider ways how we're going to search through the broad number of agents. And so really we're investigating the condition. We're not focused on individual agents. Putting these kind of things together, you have to do this right, but they get gains through sharing controls, the adaptive features within them, and the ability to invest those savings forward. And these new remap trials, one of which is being tried in COVID, also consider this as adding factorial features if you have different agents that can be combined. All right, I think I'm finished. So happy to take questions or whatever needs to be done.
Thank you so very much. This was a fabulous presentation. I really appreciate your thoroughness and even some of the comic relief throughout. I do notice that we did have a chat question from Dr. Adderwover. He wrote the question around slide 36 when you were talking through the issues of sample size. And so his question is, what adjustments, if any, are made during comparison of treatment and control groups, given that these groups may differ at baseline for adaptive trials, since treatment groups may not necessarily originate from the same population as the controls?
So in this case, there is randomization between all of the arms. So the baseline characteristics should be equalized by the randomization at baseline. Certainly the analysis model can contain covariates to do further, either to just account for that variation or to potentially account for differences. But usually the randomization does create baseline equality.
We probably have time.
Now, sorry, if you're asking a separate question here is how might you compare an agent that was early in the trial versus one that happened, say, a couple years later. That we tend to either only focus on concurrent control arms, for example, or we may do time varying models. That's actually an active area of research at the moment.
And probably our last question we'll have time for. Dr. Kern asks, can a platform trial be performed with historical controls if there's a long history of the standard of care?
Yes, so you can bring that in. What I would actually recommend, so I do a lot of research in this kind of area as well, I'd recommend that you enroll controls just to make sure that your historical controls are on point. What iSpy 2 does is it actually has the entire 10-year history of the control arm within iSpy. And so there's a model that keeps track of it over time, and that whole time course is brought in. That's one of those bells and whistles that gets you down. You can lower the 849 by bringing in that kind of historical information, either external to the trial or the internal past history of the control arm within the trial.
Okay, with that concludes the seminar. Thank you again, Dr. Viele, for coming and presenting your work and talking to us about adaptive trial design. If you would like to give a reaction to thank our speaker, I'm sure if there are other questions that you have that you'd like to ask and follow up later, please let us know. And again, this recording will be posted on the CCTS website.
Let me mention one other thing. So if anybody has questions, you can contact me on Twitter. And my email is just, you should be able to find that on Berry Consultants' website. I'm happy to ask.
Thank you so much.